Hello, my name is Priyanka Arora. I am from the Department of Biotechnology, Rama University. I am here to deliver a lecture on PCR. PCR, as the name suggests, it is a polymerase chain reaction. Polymerase means the enzyme which we are taking, the DNA polymerase or the Taq polymerase or any kind of polymerase which is required for reaction or amplification of the DNA. Okay. Chain is the polymerase chain reaction. The chain is the number of cycles, it is all the steps are repeated in a number of cycles, that is it is a chain reaction. Okay. So, it is also known as in vitro gene cloning, or the gene cloning method is for the cloning of the DNA which is happening inside the vector and the host. This type of gene cloning method is happening in the laboratory. Okay. So, it is a method of amplification of the DNA. Okay. So, in the experiment, if we require the more amount of the DNA and the initial DNA is very less, this is a method which is required to amplify the amount of DNA. Okay. Earlier, the DNA amplification technique is carried out using the cloning method, in which we require the DNA which is to be amplified, then it has to be inserted in the vector has to be transformed into the host organism, and after that we will give the condition for the cloning to happen, and out of which the DNA or the cloned product is harvested from the cell. Okay. So, this is a very tedious and a long process. To simplify this process, this kind of amplification techniques is carried out using a PCR. Okay. This PCR was first reported by Carey Mullis in 1983. Okay. In this experiment, what we do is we take the DNA which is to be amplified, we create a reaction mixture and use it in a machine. This machine is known as thermal cycler. It is known as thermal cycler because this is a temperature variation is very important in the PCR reaction. Okay. So, it is carried out in cycles. So, the temperature variation which is carried out in a cycling technique is carried out in a thermal cycler machine. Okay. So, this is the easier procedure for the amplification of the DNA. Okay. So, if we take the amount of the DNA, if this is the amount of the DNA, if we take some initial amount of the DNA, what will happen is, first it will denature, it will produce a single stranded DNA. Okay. Then after that, it will extend that DNA in the first cycle. It will make the copy 1 DNA into 2, it will produce 2 DNA into 4. So, it will amplify with every step exponentially. Okay. So, this is a cyclic amplification. So, the amount of the product which we require will depend upon the initial amount of the DNA which we have, or for the number of cycles this reaction has been carried out. Okay. If we talk about the vector experiments, using the vector, the amount of final product which we get is a variable factor. Okay. It depends upon the initial DNA, yes. It also depends upon the vector which we are using. The copy number of the vector is very important, and the host also, it depends upon the host also. So, the amount of the DNA or the final product which we are getting is a variable. But in this case, in the PCR, what is happening is we are getting the amount of final product as per our requirement, because this is happening in a machine. Okay. This is happening in the laboratory purpose. Okay. So, how this thing happens is, we will see the steps of it. First step is denaturation of the DNA. What is denaturation of the DNA? Denaturation of the DNA is the breakage of the hydrogen bonds between the DNA strands. Okay. So, this idea of PCR has been developed from the DNA replication which is happening in the body. So, what is happening in the DNA replication is, if we have the initial set of the DNA, first using the helicase enzyme, this DNA set is denatured. Okay. Then this is 5 prime, 3 prime, 3 prime and 5 prime. Then the primer is developed, DNA dependent RNA polymerases, the primer is developed, and after that this primer is extended. We know that we require the primer because we should have free 3 prime OH. We should have free 3 prime OH, okay, whose extension is required to create the copy of the DNA. Okay. So in this was happening in the replication or inside the cell. In the lab, what is happening? First, we have to denature the DNA because we require the single stranded of the DNA. For that, since we cannot use the enzyme, the helicase which is present in the cell, what are we doing is we are using the temperature. The temperature of 95 degrees Celsius or more is required for denaturation of the DNA. Okay. At 95 degrees Celsius, what is happening? The hydrogen bonds between the two strands of the DNA gets broken and the DNA is converted into two single strands. Okay. After that, since in the replication what we require the primer. Here also we are requiring the primer. We are designing the primer which is complementary to our DNA template. Okay. And that DNA template, we are designing two sets of primer because now we have two strands of the DNA. Okay. We require the forward primer and the reverse primer, and it will anneal to our DNA, anneal or bind to our DNA template. So the next step is annealing. Okay, which is happening, the annealing or the binding of the primer at our DNA template is happening at 50 degrees Celsius to 65 degrees Celsius. This range of temperature is the best that works for the annealing of the primer. There is a range of the temperature because the annealing temperature depends upon the nucleotide content of the DNA or the primer. Okay, the next step is extension. The primer which we have designed needs to be extended for the full replication or to form the full copy of the template DNA which we have. For that, we require the extension. This extension happens at 72 degrees Celsius. Okay. So, we will see why this extension happens at 72 degrees Celsius. For this, we will see how the master mix or the reaction mixture of the PCR is created. First is we require a DNA template, the DNA template whose amplification is required. Okay, we have two sets of primer, the forward primer, the reverse primer, from which the amplification or the extension will happen. We require DNTPs. DNTPs are the nucleotides. We have four set of nucleotides A, T, G and C, which is required as a raw material because the nucleotide has to be added. Then we have the polymerase, which is the enzyme. The polymerase enzyme is the enzyme that catalyzes the formation of phosphodiester bond between the previous DNA and the incoming nucleotide. Okay. For this reaction, we have seen that in the DNA, in the replication which is happening inside the cell, if we see the prokaryotic or the eukaryotic systems, we have seen that generally this replication is happening at 37 degrees Celsius. So, the enzymes are more active at 37 degrees Celsius. Okay. So, at 37 degrees Celsius, the DNA polymerases which is present in the body or the cells are active. It will increase the temperature from 37 degrees Celsius for the denaturation of the DNA template, then what will happen is our enzymes will get degraded. So, for that the DNA polymerases which we are using in the laboratory is isolated from bacteria which is functional at very high degrees Celsius. Okay. So, this bacteria Thermus aquaticus is the organism which is hot spring bacteria, and it is able to replicate its DNA at 95 degrees Celsius or so. Okay. So, the Taq polymerases are taken from Thermus aquaticus, so that these are heat stable polymerases. Okay. So, that it can withstand the temperature variations and it remains stable. And last what we required is the buffer for the reaction to happen. There are many type of DNA polymerases, not only the Taq polymerases but other organisms are also present which are stable at very high temperature. Okay. They have different temperature at which they perform their work. The Taq polymerase is most functional, or the polymerization activity of Taq polymerase is at 72 degrees Celsius. That is why the extension temperature is 72 degrees Celsius. Okay. If we will see how this DNA polymerase works, or how this PCR work is, we have the initial set of DNA which is 5 prime 3 prime. First is denaturation, which happens at 95 90 to 95 degree Celsius. So, at this temperature, first cycle of denaturation happens, in which we get two single stranded DNA, which is 5 prime 3 prime and 3 prime to 5 prime. Next is annealing. The annealing is at which the correct primer is attached to our DNA template. Okay, we have a forward primer, we have a reverse primer which is attached to our DNA template. This is happening at 56 degrees Celsius to 65 degrees Celsius, and next is extension, which is happening at 72 degrees Celsius, and in which this DNA primer is extended and we get the replicated or the two sets of DNA from a single copy of the DNA. This is happening in the first cycle. So, this is the first cycle. We can repeat this cycle for 20 to 40 times to get the amount of final product as per our requirement. Okay. If we talk about the PCR, the most important factor that is required for the successful PCR to happen or the successful amplification of the DNA is the primer designing. Okay. It is the most important factor, because if the correct primer is not binding, then there is no amplification of the DNA. Okay. For the primer designing, the most important thing is the size of the primer which we are designing. The size of the primer should be 18 to 22 nucleotide long. Okay, 18 to 22 nucleotide long. It should not be more than, as per the experiments conducted, it has seen that if the primer is more than 22 nucleotides, it will promote non-specific binding at this places which are not very specific to our required DNA segment to be amplified. If it is less than 18 nucleotides, then it will show its low sensitivity. Okay. Next is if the GC content of the primer. The GC content should be 40 to 60%. Why we are considering GC content of the DNA is because we know that G binds to C with 3 hydrogen bond and it is a very strong bond. Okay. To break the GC, we require more amount of heat or the more amount of temperature. Okay. That is why we have, for the proper or the stable binding of the primer in our DNA, we require good amount of GC content. Okay. But if we will increase the GC content by more than 60%, what will happen is it will require more melting temperature for it to be denatured, and it also causes the mismatching will happen more. Okay. Next is there should not be complementarity between, no complementary sequences should be present between two primers, because if there is a presence of two complementary sequences, what will happen, the forward and the reverse primer will form a primer-primer dimer, and hence it will not bind to our template. Next is there should be no repeated bases. If we have repetitive bases in our primer, what will happen? They will bind to each other, they will form the hairpin loops, so that they will remain inactive and it will not bind to the template. Okay. Last is that annealing temperature should also be taken care of. The temperature at which it binds to the DNA template should also be taken care of. It should not be more than, it should be in the range of 56 degrees Celsius to 65 degrees Celsius. Okay. And we have to take care that it should be 5 degrees less than the melting temperature. Okay. 5 degrees less than the melting temperature. What is the melting temperature? Melting temperature is the temperature at which half of the DNA strands gets denatured. Okay. The annealing temperature should not be more than melting temperature. If it will be more than melting temperature, then before annealing it will melt and it will get denatured. So, these are the key points which is required while designing the primer, and primer designing is very necessary for the proper PCR conduction. Okay. With this I end the lecture. Thank you so much.